Hi friends, good afternoon. How are you? Hope all are fine. Today, Holi is observed by crores of people throughout the world. Holi is known as the festival of spring, the festival of color, and also as the festival of love. Holi celebrates the eternal and divine love of Radha Krishna. It also signifies the triumph of good over evil. I wish all of you a happy and colorful Holi. Welcome to Sam online class. This is Dr. Subrat Kumar Mahanti. Today we shall discuss about thymus. Thymus is an encapsulated primary lymphoid organ that is responsible for maturation of T lymphocytes. This is a bilobed organ and it is located behind sternum between two lungs. In this diagram, these are the two lungs. This is right lung and that is left lung. And between these two lungs, here you can see the presence of thymus. And thymus, it is a bilobed organ. One is right lobe and another is left lobe. These two lobes are connected at the middle by a connective tissue isthmus. So now let us discuss in detail about the structure of thymus. Thymus is an encapsulated primary lymphoid organ. What is primary lymphoid organ? The lymphoid organ at which immune cells develop from immature precursor is known as primary lymphoid organ. So thymus is a primary lymphoid organ. It is surrounded by a connective tissue capsule. This is the connective tissue capsule that enclose the thymus. And the two lobes of the thymus are connected at the middle by a connective tissue isthmus. Histologically, thymus is divided into subcapsular cortical region and medullary region. Inside the thymus, there is little supporting connective tissue. And the cortical region is darker than medulla. This is due to the presence of a greater number of lymphocytes in the cortical region as compared to medullary region. And the connective tissue capsule gives rise to septa. These are the septa. And the septa divide the thymus into interconnecting lobules. These are the interconnecting lobules. So the capsule gives rise to septa that partially subdivide thymus into interconnecting lobules. And the capsule consists of two layers, outer layer and inner layer. Between these two layers, there are occasional clusters of lymphocytes. And the inner layer of the capsule invaginate to form the septa. And the septae partially divide the thymus into interconnecting lobules. From the septa and also from the capsule, fine trabeculae originate and they penetrate deep into the center of the lobule. Among all lymphoid organs, thymus is unique because it is an epithelial organ. We are discussing about the structure of thymus. Thymus is a primary lymphoid organ. It is also a epithelial organ. Histologically, thymus is divided into three different regions. One is subcapsular cortical region, second is cortical region, third is medullary region. Thymus contain two major categories of cells. One is epithelial cells which provide a framework. Second is thymocytes. Thymocytes are the immature lymphocytes that has to undergo maturation in the thymus to form T lymphocytes. So now let us discuss about the structure of cortex, medulla, blood-thymus barrier, and we will also discuss about the types of cells that are present in these regions. So first is cortex. Cortex is present below the capsule. It contains densely packed small and immature lymphocytes. It also contains a transient bone marrow derived population of phagocytic macrophages, epithelial cells and dead cells. The macrophages are phagocytic cells that can phagocytose the dead cells as well as any foreign substances that may enter into the thymus. The cortex contains three types of epithelial cells. One is squamous thymic epithelial cells. These epithelial cells are important in the formation of thymus blood barrier. This thymus blood barrier, its main function is to prevent the entry of foreign antigen from vascular system into the thymus. Second is stellate thymic epithelial cell. These epithelial cells form the cytoreticulum. Next is other squamous thymic epithelial cells that are also present in the cortex. These cells form the corticomedullary barrier. Corticomedullary barrier is also known as corticomedullary junction that is present between cortex and medulla. So what is corticomedullary junction or corticomedullary barrier? Corticomedullary barrier separates the outer cortex from the inner medulla. This barrier is characterized by plentiful of blood vessels, mature and immature T cells, dendritic cells, and variable number of B cells and plasma cells. This is about corticomedullary barrier or corticomedullary junction. Second is medulla. Medulla is the inner zone. It is less densely populated with various types of cells. Medulla contain more mature T cells, prominent epithelial cells, Husserl's corpuscle, macrophages, dendritic cells and B cells. What is Husserl's corpuscles? Husserl's corpuscle are concentric congregation of epithelial cell that release cytokinin, and cytokinin regulate dendritic activity. The medulla contain three types of epithelial cells. One is squamous thymic epithelial cell, stellate thymic epithelial cell, and Husserl corpuscles. And Husserl corpuscles are the concentric congregation of thymic epithelial cells that release cytokinin, and the cytokinin control or regulate dendritic activity. Both the cortex and the medulla also contain other non-thymic, non-lymphoid cells. So the non-thymic, non-lymphoid cells that are present in the thymus are: one is myeloid cells. The myeloid cells include monocytes, macrophages and dendritic cells. Second is fibroblasts are also present. Fibroblasts are the cells that facilitate the production of collagenous matter. This is about the structure of cortex, medulla and corticomedullary barrier. Next we will discuss about blood-thymus barrier. After that we will discuss about the function of thymus. Although the thymic epithelia allow for the exchange of small molecules via their high density of membrane transporter and intercellular junction, they don't allow for the exchange of large molecules including foreign antigens. In the cortical region of thymus, there exists a barrier known as blood-thymus barrier. Blood-thymus barrier consists of several layers of different types of cells that prevent the entry of macromolecules and foreign antigens into the parenchyma of the cortex. So let us know about the blood-thymus barrier. The blood-thymus barrier is made up of several different layers of cells that prevent the transport of macromolecules from the vascular space into the parenchyma of the cortex. But the blood-thymus barrier allows the entry of pre-T cells so that they can undergo maturation in the thymus. Then let us know about the function of thymus. The function of thymus is maturation and education of T lymphocytes via positive and negative selection. The T lymphocytes originate in the bone marrow, and these T lymphocytes move from bone marrow to thymus via blood. In the thymus, the pre-T cells undergo maturation and education. So the thymic cortex contains immature and undifferentiated T cells that first build a T cell receptor via a BDJ recombination. The T cell receptor signal from the cell surface via a CD3 signaling complex that result in the upregulation of both CD4 and CD8 co-receptor molecules. These T cells are known as double positive thymocytes. These double positive thymocytes possess both CD4 and CD8 co-receptor. These T lymphocytes undergo two types of selection in the thymus. They undergo positive selection in the cortex and then migrate to medulla, where they undergo negative selection. So let us know about positive selection. T lymphocytes first undergo positive selection in the cortex. In this process, T lymphocytes that are unable to recognize self-antigen presented by MSC1 or MSC2 molecules are targeted for apoptosis. So, the cells which are unable to recognize the self-antigen presented by MSC1 or MSC2 molecules undergo apoptosis. The T lymphocytes that successfully complete positive selection, they retain only one co-receptor, either CD4 or CD8. These cells which possess either CD4 or CD8 co-receptor are known as single positive T cells. The single positive T cells then migrate to the thymic medulla, where they undergo negative selection. In the process of negative selection, T lymphocytes comes into contact with self-antigen presented by macrophages and cytoreticular thymic epithelial cells. If the T cell receptor of T lymphocytes bind these antigens too strongly, then the lymphocytes are targeted for apoptosis. So, the T lymphocytes which bind these antigens too strongly undergo programmed cell death. The process of positive and negative selection are highly specific, and during this process, about 95% of the cortical thymocytes undergo programmed cell death. This process, this pathway explains the process of positive and negative selection that occur inside thymus. So first we will see that the pre-T cells, they first build their T cell receptor by BDJ recombination. Then the T cell receptor signal from their surface via a CD3 signaling system that result in the upregulation of CD8 and CD4 co-receptors. So the cells are known as double positive T cells because they possess both CD4 and CD8 co-receptors. These double positive T cells undergo positive selection. Those cells which successfully complete the process of positive selection, they then proceed through negative selection. So after negative selection, we will get two types of T cells. One is helper T cell and other is cytotoxic T cell. The helper T cell retains CD4 co-receptor and the cytotoxic T cell retain CD8 co-receptor. Hi friends, this is about the structure and function of thymus. Please share, like and subscribe my video. Thank you one and all.